Chromatography is the collective term for a set of laboratory techniques used for the separation of mixtures. The mixture, in other words, the sample, is carried by a liquid or gas and is then separated into its respective components as a result of the different affinities of the components as they flow through a stationary liquid or solid phase known as a column. Three of the main techniques of analytical chromatography analysis are gas chromatography, high-performance liquid chromatography, and ion chromatography. So to define chromatography in simple terms, it is the separation of compounds based on their affinity for a bonded stationary phase. Of course, there are several other forms of chromatography. Of particular interest in the life sciences and biological fields are gel filtration, ion exchange, and affinity chromatography. However, the basic principles remain the same for all of these. So, why perform analysis by chromatography? It is a very accurate and selective methodology with literally an endless set of applications. It is versatile and reproducible. All chromatographic systems have an injection system to introduce the sample and a column which contains the stationary phase which analytes bond to. A mobile phase carries the analyte to and through the column, and it also has a detection system to detect the analytes as they elute selectively from the column. All modern high-quality systems can be hyphenated or joined together with mass spectrometers or other devices depending on application. The applications of chromatography are endless, as we have said, but some examples include environmental, pharmaceutical, food, residue testing, drugs of abuse, petrochemicals, and forensics. Like almost all analytical techniques, gas chromatography and high-performance liquid chromatography are comparative. That means that the instruments must be calibrated with a known, the standard, and this is compared with an unknown, the sample. In general, because chromatography is a sensitive, accurate, and relatively sophisticated technology, the knowns usually take the form of a full calibration curve. There are also instances where other types of standards, such as internal standards and surrogate standards, are also used. All of this is explained in several other Reagecon publications. When you take into account that the range of applications of chromatography is virtually limitless in terms of both analyte and industry, it is natural to expect that the range of types of standards available is extensive, and these are often classified as organic standards. Reagecon is at the forefront of producing organic standards, and our offering is not only enormous but growing exponentially. Reagecon produces a vast array of organic standards in many formats. These include very commonly used standards such as volatile organic compound standards, phenol standards, polycyclic aromatic hydrocarbon standards, and pesticide standards. The range also includes less widely used but equally important products such as phthalate standards, azodimetabolite standards, fatty acid methyl ester and fatty acid ethyl ester standards, nitrosamine standards, polybrominated biphenol standards, polybrominated diphenol standards, and other flame retardant standards, polychlorinated biphenol standards, phthalate standards, and semi-volatile organic compound standards. Finally, let us comment briefly on the features and benefits of Reagecon's organic standards. We have an extensive range of organic compound mixtures and single compound standards available, which can be used with a wide variety of instruments including GC, GCMS, HPLC, and LCMS. Our range mirrors key standards required for EPA methods, and our offering is also positioned to supply standards missing from competitor ranges. Reagecon's chromatography standard test methods are ISO 17025 accredited and presented in high-quality amber ampules. There are many customised options available. More detailed technical information and specific product information can be obtained at www.reagecon.com, either through our web shop or through our knowledge centre.